All right, hello everybody. We are going to talk about chromosomes today. We're going to talk about chromosome linkage and chromosome maps. I'm going to split this particular Google slide presentation into two different videos because it's just going to get too long, so it'll be easier to take in little bite-sized pieces. All right, let's go ahead and get started. So first of all, I just want you to take a look at some of these statistics and see if any particular questions pop up. Cystic fibrosis, just take a look at it. I'm not going to go into a whole lot of detail on it. Sickle cell anemia, look how many people come down with it, what populations are more likely to come down with it. Blue-yellow color blindness, ever heard of that? Fairly rare. Anything come up with that? Red-green color blindness. And then hemophilia. Anything noticeable about hemophilia? So why are some populations more likely to have certain genetic traits than others? So we're going to learn a little bit more about that, like why certain ethnicities or cultures or races are more likely to have particular traits. We'll learn more about that actually in our natural selection notes in our natural selection unit. But right now, I'm hoping you're wondering, why are males more likely than females to have certain traits? And we're going to get to that. So give me a second to kind of lead us down that path. All right. So way back when we were first learning about genetics, there was a researcher whose name was Thomas Hunt Morgan, and he studied fruit flies. And he studied fruit flies because they're a model organism. And still today, we study fruit flies. Fruit flies have very few chromosomes. They're super easy to grow, reproduce. They have some really obvious traits, like the red eyes in the photo that I have on every slide here. They have white eyes, red eyes. Their bodies come in different colors. They have wings that come correctly shaped and misshapen, all kinds of things. So he did a whole bunch of experiments with hundreds and thousands of different fruit flies because it was very easy to breed them. So his team, Morgan in particular, bred red-eyed females with white-eyed males. And I have the Punnett square down there. So W plus, the plus sign means wild type. And the wild type is the more common trait. And in this case, it's the dominant trait. W means white, and that's the recessive trait. So he crossed the original parental generation. That's what the Punnett square is down at the bottom, the green one. The original parents, the females were homozygous dominant and the males were homozygous recessive. And he crossed them, and unsurprisingly, all of the offspring had red eyes, just the way we expected. Let me get my picture out of the way here. The F1 were all red-eyed. It's sensible because red is dominant, and so we see that in the Punnett square here. All of them were red. They were also all W plus and W, so all of them were heterozygous. And it's exactly the way Mendel expected it, and it's exactly the way you do a regular Punnett square. But then things got interesting. So then they took the F1 generation and they bred them. The F1s are the ones that are all heterozygotes. So here's one parent that's a heterozygote. We took a female heterozygote here, and we've got a male heterozygote here. And so we predict that 3 out of every 4 will have red eyes and 1 out of every 4 will have white eyes. So that's our prediction. What he actually got was super surprising. All of the female in the F2 generation, so look at the results on that diagram, all of them were red. And half of the males were red and half of the males were white. That's not exactly what he had predicted. The F2 did have the expected 3-to-1 ratio. There were 75% red eyes and 25% white eyes. But when you do the math, you should still have 25%, that 25% that have white eyes, half of them should be females and half of them should be males. And it didn't work out that way at all. There were no females with white eyes. And so that was a little bit of a head scratcher. So the conclusion of the researchers, my picture's kind of in the way. I got to find a good place for it. So there's the F2 generation with the 3 red eyes, the 3 that had red eyes, and the one that had not 3 red eyes, and the one that had white eyes. So their conclusion was that the eye color gene must be carried on the X chromosome. And the bent Y chromosome, let's look at it, does not have the color gene. So here's the P generation. This is the original parents. The female has 2 X chromosomes, and she has the dominant trait on both for red eyes. The male has 1 X chromosome, and he's got the recessive trait for white eyes. And then his second chromosome is the Y chromosome in the sperm that does not carry a color either way for eye color. So her eggs, they only show one example of her eggs because they're homozygous. No matter what, her eggs are always going to be the same. She's going to carry a W plus gene in all of her eggs. So they just show you one example of her eggs instead of doing a whole Punnett square. His sperm, half of his sperm will have the recessive white gene, and half of his sperm will have the Y chromosome. So when these sperm fertilize these eggs, you could have females that had red eyes, and you could have males that had red eyes. So to Hunt Morgan, who's looking at these, they all had red eyes. So nothing was surprising. It was exactly the way the Punnett square said it should be. He can't look at the chromosomes. He can't see the chromosomes and see that the males are getting this Y chromosome and that it's missing a color gene. He can't see that. All he sees is a whole bunch of red offspring, exactly how he predicted. It's not until we get to the F2 generation where things went funny. So let's see why they went funny. So the females now are heterozygotes. The F1 females are heterozygotes. So now their eggs can be either dominant red or recessive white. So that's why we've got two pictures here. The male's sperm still can either be the one that colors the X chromosome that gives them the colored eye, the eye color, or the Y chromosome. I keep wanting to call it a sperm. It is in the sperm. Anyway, so the males now can donate a red or they can donate a Y chromosome. So they're going to donate a Y chromosome to all the boys. So the boys are always going to get just a Y chromosome from their dad. So the only color chromosome is coming from their mom. Their mom was heterozygous. So half of the eggs are going to carry the red gene and half of the eggs are going to carry the white gene. So that means that half of the boys will be red, have red eyes, and half of the boys will have white eyes. With the girls, they all inherited for sure dad's red X chromosome. So no matter what mom gave them, because he was donating that dominant X, the dominant red trait on the X chromosome, all the girls' eyes appeared red. So Thomas Hunt Morgan figured out, well, these must be carried on the X chromosome, and there must be no gene for eye color on the Y chromosome. And that's why we're getting these interesting proportions. So it says in the F1 generation, everyone looked red because they inherited mom's dominant W plus. So that's why they all looked red. And then get my picture out of the way. Importantly, the F1 generation females were heterozygous. Here they are. Right there, they're heterozygous, but the males were not. The fathers did not have a color gene to pass on to their sons. So the fathers of the F2 generation, here they are, this is their sperm, always passed red genes on to their daughters only. And then they just passed on the Y chromosome to their sons. The sons received a red or a white from their mothers, and that's what determined their eye color. All right, so let's just go through a couple quick things. Sex linkage, I just want to show you this image because it varies from species to species how sex is determined. So you're familiar with the XY system in humans, and there's actually lots of organisms that share the XY system. In grasshoppers, we have the XO system, and O in this case is like an empty set. So the females are XX and the males have just an X. There's not a Y chromosome. I'm going to skip over to the bumblebees there. The bumblebees, just interestingly, the females are diploid and the males are haploid. So they all have one of every chromosome, whereas the females have two of every chromosome. And then in the chickens, and this one shows up on an AP test, the chickens have what's called the ZW system. So the females this time are the heterozygotes. They have a Z chromosome and a W chromosome. The males are the homozygotes. They have two Z chromosomes. So compare that to humans. Under normal circumstances in a human, females are XX. We're the females, I'm one, are the homozygotes. The males are the heterozygotes, but in chickens, it's the reverse. So the roosters are heterozygous. Okay, moving on. So it says here, let me get out of the way again. So why do males show sex-linked traits at higher rates than females? Well, number one, if it's carried on the Y chromosome, only males get that trait, so males are going to show any traits that are carried on the Y chromosome, they're going to show it. Number two, females, if it's carried on the X chromosome, females have two copies of the X chromosome, and I like to think of it as sort of like they have a backup copy. So if one copy of the X chromosome has something wrong with it, the other chromosome can be used. So it's kind of like a backup copy. Boys don't have that backup copy, so if they have a chromosome that's not working right, that's the one they got to use. So now the last thing that I will just mention, because it adds a little twist in here. But early in development, so females have two X chromosomes normally. Early in development, one of the X chromosomes just is inactivated. It stops working. So we're going to use the one that's functioning better. So one of the chromosomes stops functioning, and that one that stopped functioning, it's now called a bar body. And that just means that it's there still, but it's not working. It's not being read, and its genes are not being interpreted, and proteins aren't being produced from it. And I wrote in this little note here, it says every extra X chromosome in a cell becomes a bar body. It is possible to have numerous X chromosomes, and all of them will shut down except for one. One should always be active, and the rest would become bar bodies. All right, moving on. Just a quick reminder about how to do a sex-linked problem. If you have any trouble doing sex-linked problems, pause this, let me get out of the way, and take a look and just make sure. What's super important is you have to show with these kinds of Punnett squares, you have to show the sex chromosomes. So you have to indicate the male is XY and the female is XX. Do you need to draw the little spermies or the eggs? No, you don't need to draw that, but you do need to make sure you have X's and Y's. The reason you need to do that is that males only have the trait, in this example, on the X chromosome. So notice every time that the Y does not have a little letter, the actual gene, it doesn't have, or the allele, it doesn't have that next to it. So the Y is like blank. So pause this and look it over if you need to. All right, and then sex-linked pedigrees. This is a pedigree where the trait is carried on the X chromosome, so we call it an X-linked, and I'm telling you that it's a recessive trait. So what are the genotypes of the following individuals? And so I want you to pause this video if you haven't seen this already, and try to figure out what are the genotypes of those particular individuals. I'll talk you through the first two, and then you can figure out the rest. Number one is a female. Females have curves, maybe as a way to remember that. And so females are XX, so you need to show the two X's. And then because she has this recessive trait, she must have little n's. So number one is X little n, X little n. Her partner, a male, is X, and he's normal, so he's X big N. And then by the Y, we don't put anything. So just X big N Y. And then go ahead and pause this video to solve the other ones. I'm just going to go ahead and so pause it right now. And now I'm going to go ahead and show all the answers for the other ones. And let me know if you have any questions about them. All right, now let's jump into gene linkage. This is kind of the meatiest part of the notes that we're going to be talking about. So let me once again get my image out of the way. So gene linkage, some genes are inherited together because they're found on the same chromosome. And so we say that they are linked. So you can see an image of A, B, and C. They're linked. They're on the same chromosome. So there were some puzzles trying to figure out what linkage was and explaining some of the results. So let's take a look at this puzzle. So this is also Thomas Hunt Morgan's work with his fruit flies. So here he's got a female that is a dihybrid. It's the F1 dihybrid. It doesn't matter. But she's heterozygous for both the color of her body and the shape of her wings. So in this case, B plus is the dominant gray body, and VG plus is the dominant long, full wings. Little b stands for black. The lowercase b without the plus stands for black, and it's recessive. And the VG stands for vestigial wings, which are wings that are really small. They're kind of, they're not shaped correctly. So vestigial wings. So she's a heterozygote, and so he is doing a test cross. He can't tell that she's heterozygous. She could be homozygous dominant, and he can't tell. So he's going to cross her with a male that is completely recessive. He can see that the male is recessive because he can see the black and he can see the vestigial wings. And the only way to be recessive is to have both copies. So little b, little VG, little VG. So he does a test cross with her, and he's got some ideas about how the results are going to be. So let me get out of the way. I'm always in the way, people. Okay, so if the genes, these are his ideas, if the genes are on different chromosomes and inherited separately, this is like a dihybrid problem, like a typical dihybrid problem that we would do in class. You multiply the probabilities. So I've made the two Punnett squares for you. Notice that the female here, she's B plus and B, so I've got her right here, B plus and B, and then the male is little b, so there I've got him there, and then the VG plus and VG, so it's two monohybrids multiplied together. So what would we predict the results of their offspring would be? What would his predictions be? Well, if we multiply them, let's try. If these two boxes are the same, and that would produce gray body. If it's got a plus, it would produce a gray body. And these two boxes, if it's got a plus, it produces normal wings. So we multiply them. Half of the boxes would make a gray body. Half of the boxes would make normal wings. So 1/2 times 1/2 is 1/4. So we would predict that a quarter of the offspring would be this one right here, gray and normal wings. So then if you keep doing the multiplication, it's half and half in both of the Punnett squares. So we would actually predict a quarter of the offspring will be gray normal, black vestigial, gray vestigial, and black normal, a quarter of each. So we call that a 1-to-1 to 1-to-1 ratio. So that's one prediction that he made. And so that was one possibility. Another prediction he made was that they were actually on the same chromosome and these genes are going to be inherited together. So she has B plus VG plus on one chromosome, and either she got it in her egg or she didn't get it in her egg. And then she also has a B and a VG, and it's either in the egg or it's not in the egg. The male, his sperm are always going to have the lowercase letters. So it's the same BVG here and BVG here. So if you cross them and they stay stuck together, you're going to have a gray normal and a gray normal 50% of the time. And we're going to have black vestigial and black vestigial 50% of the time. So in other words, they're going to look like their parents all the time. Half of the time they'll look like the mom and half of the time they'll look like the dad. So that's his other prediction. That's his other idea. So then the question is, what were the results? Were these linked on the same chromosome or were they on separate chromosomes? So the actual results are kind of unclear. What's really going on here? So instead of seeing a 1-to-1 to 1-to-1 ratio, which would indicate that they were not on the same chromosome, he didn't get that. Instead of seeing a 1-to-1, 0-to-0 ratio, indicating they were linked on the same chromosome, he didn't get that either. So what's going on? And hopefully you know kind of where we're going with this. So gene linkage, it says, notice that linked genes can be separated. They're normally together, but they can be separated during crossing over events in prophase 1 of meiosis. Look at the chromosomes. A and C are farther apart. So where crossing over occurs is completely random. So the chromosomes break and they trade little sections of DNA. And one time the crossing over, so if I have, let's say here's A and here's C and B is in here. The crossover might occur here one time, it might occur here one time, it might occur here one time, here. But it's always separating the A and C because they're fairly far apart. Genes that are separated onto different chromosomes and are no longer linked can be inherited separately. So we're starting to try to understand, why did Thomas Hunt Morgan get these weird results? Almost 1,000 individuals that were like the mom, almost 1,000 individuals that were like the dad, but then 200 that were like neither parent and 180 that were like neither parent. How did that happen? Crossing over is where we're going with this. So moving on. So we're going to start making now what are called chromosome maps, and Thomas Hunt Morgan is what's leading us to this. So it says we can use the percent of time two traits are inherited together or separately to determine how far apart they are on the chromosome. If they're far apart, they're going to cross over frequently and be inherited together infrequently. If they're close together, then they're going to be inherited frequently. So we'll get to the maps in just a second. So let's take a look at this diagram. This is the same exact situation that we had before. We've got the gray female who's heterozygous, and she's being test crossed with that black vestigial male. And I want to remind you really quickly, this is called autosomal linkage instead of sex linkage, because this is taking place on chromosomes that are not sex chromosomes. So the male has 2 and the female has 2. It's not 1 and 2 or anything like we talked about previously. Okay, so let's take a look at this diagram. Let me get my picture out of the way again. So we've got the gray female, and she's got this red chromosome that's got both of the dominant traits, and she's got this pink chromosome that's got both of the recessive traits. Now watch what happens. During meiosis 1, they cross over, and 2 of the chromosomes now look a little bit different. This pink one, instead of having 2 recessives, now has 1 recessive and 1 dominant. And this red one, instead of having two dominants, it now has one dominant and one recessive. So now they're going to be separated into different eggs. So in one egg, she could have this red one. There it is. In one egg, she could have this one that's mostly red with a little pink. There it is right there. In one egg, she could have this mostly pink with a red. There it is. Those are called the recombinant ones because during crossing over, they recombined. Or her egg could have the original recessive chromosome, and there it is. Now, the male, it looks like the male isn't experiencing any crossing over. I think that's a flaw in their diagram, like it's confusing. The reason they're not showing crossing over is that there's really nothing to cross over. It is happening. Crossing over is occurring in the male, but because he has only recessive traits, even if they cross over, it doesn't make any difference. So it is happening, but all of his sperm, there should be, normally we show 4 different sperm here. They're all exactly the same. They all have the recessive traits for both the body and the wing shape. So just one sperm is available here. All right, so it says, I'm going to kind of repeat here. The female is heterozygous for body color and wing shape. The male is homozygous for both recessive traits. When the female's chromosomes go through crossing over, the traits are separated. B, lowercase b, the recessive, is now sometimes with VG plus, the dominant normal wing shape. Because the male's alleles are homozygous, crossing over doesn't really change his chromosomes. B and VG are always still together. Okay, get me out of the way here. Whoops, I failed. Try that one more time. There we go. Okay, so it says, look at the impact of crossing over on the offspring. Without crossing over, you've seen this Punnett square that I have here. You've seen this already earlier in the presentation. Without crossing over, probability expects a 1-to-1 ratio of gray normal and black vestigial, nothing more. So if they're on the same chromosome, if they're being inherited together, that's what this B plus VG plus means, is they're being inherited together. We would expect 50% to look like the mom and 50% to look like the dad. And sure enough, a lot of them look like this mom. She's the gray normal. And a lot of them look like the dad. This is the black vestigial. However, there are now a number of recombinant offspring because the crossing over separated the mom's different genes, alleles. They were separated, and so now we have these recombinant offspring. And so it's crossing over that explains why we got these weird numbers. Now, I want you to notice down at the bottom, it says the recombination frequency. I mentioned that you can make a chromosome map to figure out how far apart they are, and then it's dependent on the percent of times that they cross over and recombine. So if we look at the total number of offspring that Thomas Hunt Morgan was studying, there were 2,300 fruit flies. Exactly 391 of them showed the recombinant traits, so the traits due to crossing over. So that's 17%. 17% of the time we got offspring that were different than the parents. So we're going to make that on our map. We're going to make that 17 map units apart. So it says, we can calculate the frequency of crossing over by looking at the numbers of recombinant offspring out of the total number of offspring. Here's a question just to chew on. What would happen to the recombination frequency if the genes were actually located closer together on the chromosome? Remember that where crossing over occurs on a chromosome is random. So if it's really close together, it might happen outside of it. So what happens? The recombination frequency would be less if they were closer together. And they're separated by crossing over less often. So if they were closer together, the percentage might be 8% instead of 17%. I just pulled that number out of my hat. Okay, this is the last slide and then we're done. So these chromosome maps, how we make this. So it says we can use the recombination frequencies to make a map of the chromosome. Genes that are further apart will recombine or cross over more often than genes that are closer together. So Thomas Hunt Morgan, thank you, dude, decided to make map units, the distance between genes on a linkage map, equal to the percent of crossing over. So if they cross over 17% of the time, then they are 17 map units. They're also called centimorgans after Thomas Hunt Morgan, 17 map units apart. Now, the one thing I will mention, so let me just say this one more thing and then we'll say one more thing. that didn't make sense. The color and wing genes separated due to crossing over approximately 17% of the time. So those genes are placed approximately 17 map units apart. I guess there was one more thing I wanted to say. There was one more slide. I want you to notice this is a little confusing. So there's another gene that's called cinnabar. Don't worry about it. But it recombines 9% with the body color, and it recombines 9.5% of the time with the vestigial wings, the wing shapes. So the question is, if it's in between those two, why don't the numbers add up? 17%. Why isn't 9 and 9.5? That's 18.5%. Why are they off a little bit? And I'm just going to explain that really quickly. So it says, okay, well, let me go down here first, and then I'll explain the other one. So down here, I'm looking at this one. I'm screwing it all up. The numbers on the map don't always perfectly align. That was what I was showing on the previous slide, due to double crossing over events. What? It can cross over twice, KJ? Yes. So the first time it crosses over, they're separated. The second time it crosses over, they get put back together again. And so that actually screws up our map units a little bit. So the percentages are sometimes a little bit different than the map units, but it's still fairly accurate. So if you have a lot of recombination, it indicates that they're further apart. Not very much recombination, it indicates that they're closer together. And then this little FYI over on this side, if the frequency of recombination is greater than 50%, I want you to have this in the back of your head because this is for sure something they're going to trick you on a test question. So if it's greater than 50%, we actually can't differentiate between genes that are far apart on the same chromosome and therefore separate often due to crossing over, or genes that are simply found on separate chromosomes. They're going to be inherited in similar ways. So once it gets above 50%, your hands go up and you go, I don't know. So under 50%, that's where we can start doing that mapping. All right, everybody, that is it. Let me know if you have any questions.